Today, let's start with the hormone which is insulin. So, what is insulin and from where it is released? So, insulin is released from the pancreatic islets of Langerhans. So, this is also called as islets of Langerhans. This is pancreas. This consists of three types of cells. One is alpha, beta and gamma, delta. So, alpha cells will release glucagon and beta cells will release insulin and delta cells will release somatostatin. So these are the three hormones which are released from the pancreas. This pancreas, you will be having many cells inside. We have three different types of cells and these are the three different types of cells. Today let's talk about the beta cells which releases this insulin. So let's see where this insulin is synthesized. So insulin is synthesized in pancreas. In beta cells, you will be having ribosomes. This ribosomes will release a protein, a typical specific protein which is called as pre-proinsulin. So this is the chain of pre-proinsulin. So you may understood why I told peculiar and special protein, because this will be having a long chain which consists of 110 amino acids. So this, by losing 24 amino acids, it will become, this is pre-proinsulin, this will become proinsulin by losing 24 amino acids. So if you see this in proinsulin, you have three types of chain. This is C peptide and this is B chain. So this have A chain and B chain, and this B chain and A chain is joined with the help of C peptide. So this is how our proinsulin look like. But we can differentiate this C peptide, this A chain and B chain, they both will combine, plus it will have C peptide. So now this is A and B chain and C peptide. Okay, now this proinsulin is now converted into insulin plus C peptide. This C peptide, why they have got differentiated, why the C peptide and AB have differentiated? Because Golgi apparatus will be proteolizing this enzyme, proteolizing this peptide, this C peptide. Okay, that's why they got separated. Now we have only A and B. But still our peptide will be in our beta cell only. So this is all happening in our beta cell. So this C peptide and A plus B chain will be in our beta cell only. So whenever we need, this beta cell will release this insulin plus C peptide into our blood streams. This is how our insulin is synthesized and it is released into our blood streams. So as I told you, this insulin consists of three chains, which is A, B and C peptide, and it consists of 51 amino acids. So in this 51, 21 are A chain and 30 B chain. So totally 51 amino acids. So this amino acids, we have similar kind of insulin in pork, beef and human being. But there will be slight differences. But when you compare our amino acids with pork amino acid, then it will differentiate a little bit. Like pork insulin will be equal to our human being insulin. So mostly if we want to take insulin from outside, we will prefer only pork. So now let's see the regulation of insulin. So per day, 1 unit of insulin is released from pancreas per 1 hour. So for 1 hour we need 1 unit of insulin. So how this insulin production is regulated? This is regulated with the help of three mechanisms. First one is chemically, second one neural, third one hormonal. So let's first see this chemical. So chemically how it is done? So this beta cell have glucose sensing mechanism. So when glucose enters into this beta cell with the help of Imagine we will zoom this beta cell. So this is the beta cell. When glucose enter into this beta cell with the help of GLUT1 transporter, we have 4 types of glucose transporter: GLUT1, 2, 3 and 4. Mostly we will use GLUT4 for muscles and everything. GLUT4 is the main transporter. And if you see into the beta cells, the glucose enters into the beta cell with the help of GLUT1 transporter. With the help of this GLUT1 transporter, our glucose will enter into this beta cell. When it is entering into this beta cell, it will get phosphorylated. This glucose will get phosphorylated. So when it is getting phosphorylated, that means it is getting activated. So how it is phosphorylated? With the help of glucokinase. Okay, with the help of glucokinase, this glucose is getting phosphorylated. So now glucose entry and the glucose metabolism will activate glucose sensors and help in production of usual, right? When it is taking this glucose, it will also produce ATP. So the entry and the metabolism of this glucose will decide how this will increase this glucose sensors, and also it will increase the production of ATP. So when this is increasing this glucose sensor and production of ATP, it will inhibit potassium ATPase, ATP sensitive potassium channels. So this will resulting in partial depolarization. Okay, this will increases the calcium influxes. So what will happen as this is increasing the glucose sensor and production of ATP, this will inhibit, imagine this is the beta cell. So this will inhibit this potassium. This is potassium channel. So this will inhibit this channel. So when it is inhibiting, there won't be any influx of potassium. So when it is inhibiting this potassium, it will also activate calcium channels. When it is activating the calcium channels, there will be influxes of calcium into the cell. So when it is increasing the influxes of calcium ions into the cell, the beta cell will exhibit exocytosis, and as this contains insulin, this will release insulin from the cell. Okay, let's see again the mechanism of this one. So this is the beta cell. Regulation thing is happening in the beta cell. So every beta cell have glucose sensing mechanism. What is this glucose sensing mechanism? Whenever it is coming, whenever they see glucose, they will get attracted. So when this glucose enters into this beta cell with the help of GLUT1 transporter, it will phosphorylate this glucose. Okay, when it is phosphorylating this glucose with the help of glucokinase, it will increase the glucose sensors, and also it will increase the production of ATP. When it is increasing this production of ATP, it will inhibit this potassium, ATP sensitive potassium channels. Okay, because it is releasing ATP sensitive potassium channels. So when these got inhibited, this will activate the calcium. When this calcium channels got activated, the influxes of calcium will increase. When it is increasing the influxes of calcium, it will do exocytosis. Already beta cell consists of, when it is synthesizing, insulin got already synthesized in the beta cell. So we have this beta cells in vesicles. So through exocytosis, this beta cells will release this insulin into the bloodstreams. So this is how our insulin is regulated chemically. Not only this glucose, insulin will get activated or inactivated with amino acids, fats and ketones. That's the reason nowadays we have keto diet. So when glucose is entering into this beta cell, it will release the insulin within 2 minutes. Within 2 minutes, this whole process will happen, and this will release the insulin into the bloodstreams. This is the first phase. But the second phase will take little longer when compared to the first phase. For example, the first phase is 2 minutes, right? And the second phase will be 3 minutes, but there won't be any third phase or fourth phase. This 3 minutes will get continued. So now our insulin is released into the bloodstreams. What does insulin will do? The function of insulin is whenever we have glucose, this insulin will store in it. Insulin is it's a bag, so this will store the glucose in it, and whenever we need, it will release. For example, now I am talking, now I am standing. This because of my insulin. I have ate 2 hours back. So insulin will store this glucose and it will release whenever I need. For example, if we are not eating any food from morning to evening, what will happen? Our energy levels will decrease, right? But the insulin, it will get sense and it will think that this poor fellow haven't ate anything. So we have to store this glucose and we will release this glucose slowly. That's the reason when we are doing fasting also, we won't lose that energy unless we are doing this fasting from so many days. That's why fasting is only for one day. If we are doing continuously, then it will harm our insulin, and insulin levels will increase also. So not only glucose, some nutrients along with glucose, when they are entering into our body, that will also help in secretion of this insulin. So what are those? So when we are giving this insulin orally, this will have more effect when we are giving through IV, because along with this insulin, when we are eating something like glucose plus when we are eating any food, that will enhance the effect of this insulin, because it will also help in the secretion of incretins. So what is this incretin? These are chemical signals which are from gut. So what is the function of this incretins? This will increase insulin secretion. So that's why we need this incretins. That's the reason, as they are in gut, every insulin, when we are taking insulin, that will go into the bloodstreams and that will start to take this glucose. But the added advantage of this oral is, even this will go, like if we are giving IV or oral, they both will go into our blood circulation only. But this oral, before going into blood circulation, the added bonus is that it will go into this gut and it will activate this incretins. This incretins, when they get activated, that will also activate the insulin. So what are these incretins which are in our body? We have 4 types of incretins. The 4 incretins are GLP, glucagon-like peptide, and GIP, glucose-dependent insulinotropic polypeptides, and VIP, vasoactive intestinal peptide, and PC, pancreasoimin cholecystokinin. These are the 4 incretins. With the help of the nutrients which we are eating, they will increase the levels of insulin. They will increase the secretions of insulin in our body. So next is hormonal. Let's see the regulation of this insulin hormonally. So if you see, this is the structure of islets of Langerhans. So in this you have different types of cells and SNE. This is the reason why we are called this islets of Langerhans, both endocrine and exocrine gland. Why exocrine? Because if you see in this SNE, you have the ducts also. So whenever we need insulin, it is released into our body, as well as when there is excess of insulin, that insulin is stored in this duct of SNE. Inside this one, you have 3 types of cells: beta cells, alpha cells, and gamma delta cells. So if you see, among all these 3 cells, beta cells are more number. So here if you see, all these orange cells are beta cells. So if you see this alpha cells, they are only 25%. Okay, and when it comes to delta cells, they are only 10%. So remaining 65% will be our beta cells only. So here all the remaining cells will also regulate the synthesis of this insulin. So how? Let's see. The first one is somatostatin will decrease the secretion of insulin and glucagon. And next one, glucagon will inhibit the secretion of insulin. How? What is the function of insulin? This will decrease the glucose levels in the bloodstreams.
So what it will do, whenever we are eating something, it will take the glucose into it and it will store it. But what is the function of glucagon? Whenever we are less in glucose, this glucagon will inhibit this insulin and it will release glucose into the bloodstreams. When there is less insulin, insulin can't take the glucose and insulin can't store the glucose in itself, isn't it? That's why glucagon will decrease the levels of insulin. When it is decreasing the levels of insulin, it will increase the levels of glucose in blood streams. Okay, that's the function. And let's see the third one. Third one, insulin will inhibit glucagon secretion. If it is inhibiting glucagon only, it can store the glucose, right? It will inhibit the glucagon. Our beta cells, along with this insulin, they will be releasing a little amount of amylin. So this amylin will also do the same function. It will inhibit the secretions of glucagon, but with the help of our brain. All these three cells, they will be beside one each other, but they have different functions and hormonally they are different. They will inhibit each other and they will make the hormones in level. For example, now we have seen glucagon will inhibit insulin, insulin will inhibit glucagon, and somatostatin, it will inhibit both insulin and glucagon. Whenever there is more glucose in our body, insulin will get activated and it will do its function. It will decrease this glucagon and it will take the glucose. But when there is less glucose in our body, glucagon will inhibit insulin and it will do its function.
So now let's see the neural regulation of insulin. So in this, our pancreas are rich with this sympathetic and vagal nerves. When these get activated, that will show effects on our glucagon and insulin. So let's see insulin only today. The first step is when there is activation of this adrenergic alpha 2 receptor, it will decrease the insulin release. How? By decreasing the beta cell adenyl cyclase. Okay. And next one, when there is increase in adrenergic, when there is stimulation of this adrenergic beta 2 receptors, then it will increase the insulin release. How? By increasing the beta cell adenyl cyclase. So third one, when there is cholinergic and muscarinic activation by ACH or vagal stimulation, then that will stimulate to secrete this insulin. How this insulin is secreted? By activation of a secondary messenger IP3 or DAG pathway. In this IP3 or DAG pathway, it will stimulate this calcium channels. When calcium channels got stimulated, they will calcium ions will influxes. When calcium ions are coming into the cell, then it will secrete this insulin. Okay, calcium ions are entering into this beta cells, then it will increase this insulin secretion. So now let's see the actions of this insulin. In this, the first action is, the overall concept of insulin is, when we take this meals, it have to convert this, it have to convert into glucose, fats, amino acids, and after converting, they have to store somewhere, right? So the concept of this is insulin will store all those things, and whenever our muscles or whenever our body parts need it, then it will release to their body parts, respective body parts. And not only that, the insulin is anabolic hormone. What is a anabolic hormone? It is a it will take a smaller units of molecules and it will construct a it will take smaller units of molecules and then it will convert into a complex molecule. For example, if we are taking this glucose, so what it will do, this anabolic hormone, so this insulin will promote, as it is anabolic hormone, it will promote the synthesis of, it will promote the synthesis of glycogens, lipids and proteins. So this action of insulin and its deficiency and its results of its deficiency, and the results of its deficiency is given in steps. So let's see the first step. So what is the function of this insulin? Insulin will help in the transportation of this glucose across the cell membrane. For example, if it have to go somewhere in the cell in our body, it needs, if the glucose want to transport into any cell, we need insulin. That is the main concept. But for our muscles, most of the time, we don't need any insulin support to take this glucose, because when we are doing exercise, it automatically with glucose transporters, it will take this glucose into the cell. At that condition, they don't need any help of this insulin. But when it is going into any liver cell or into brain, because everywhere brain can't store this glucose, right? So it definitely needs this help of insulin. Whenever this glucose is entering into any cell, it needs the help of insulin. But we do have some cells which doesn't need the help of insulin. Those are skeletal muscle, as I told you, because skeletal muscle, when we are doing exercise, only when we are doing exercise, it doesn't need any insulin, because when we are doing exercise, our muscles need that glucose. If it is not taking that glucose, they won't work, because when it is taking glucose, it is converted into ATP, which is energy. So when it is not taking this glucose, our muscle won't work. Let's see the cells which doesn't need this insulin for the transportation of glucose into their cells. The first one is liver, brain, RBC, WBC and renal medulla. So these cells doesn't need any help of insulin to transportation of glucose into its cell. And conditional skeletal muscle, because exercise, no need of insulin. If there is no exercise, then they need insulin, that's why. So when I'm talking about this brain, there will be one factor which is affecting this utilization of glucose into the brain, that is ketoacidosis. So what is this ketoacidosis? So when body starts to break down this fats, actually our body will break down each and every fat. So here, when fats are breaking down in a fast rate, for example, now I have taken a meal and I have taken with lot of ghee and everything, so my body consists of fats now. So it will start to break this fat, but when it is breaking down the fats, those are converted into ketones. So in normal condition, these ketones, they won't harm us. But what if the rate of breaking down of these fats are more? Then there will be increase of these ketones. When there are more ketones in our body, it will become acidic in blood. Mostly these ketones will travel in blood only, right? So our blood will become acidic in nature. This mostly occurs in diabetic patients only. That's why it's called as diabetic ketoacidosis. So what is the function of these ketones? Our liver will convert these ketones into fuel, like energy. But when there is more, it will causes our blood to become acidic. Okay. So this ketoacidosis will interfere with the utilization of this glucose in the brain. Then it will causes diabetic coma. Diabetic coma is a life-threatening state in which this diabetic patients, when there is too high blood glucose levels or too low blood glucose levels, they will go into unconscious state. So it is reversible when it is treated immediately, but when it is not treated in time, this may leads to death also. That is diabetic coma. So let's see the second point. So how this glucose is utilized in the cell. So first glucose is entered. So imagine this is the cell. So first glucose will enter into the cell with the help of transporters. So we have two types of transporter, GLUT and SGLTs. So this SGLTs is a sodium glucose transporter and this is glucose transporter. In this sodium, the name itself says it is a sodium dependent. Whenever we have sodium, this will activate, this transporter will activate. So why do we need this glucose transporter? Because glucose can't go into the cell by its own. For example, to skeletal system, so when it is going into skeletal system, something needs to be there for traveling, right? So these are the vehicles of that glucose. So this is sodium dependent and this is sodium independent, and this doesn't need any ATP. So this is ATP independent and this one is ATP dependent. So let's see the GLUT1, where this GLUT1 is located, where this transporter is located, and what is the function of it. So GLUT1 is located in, forget about this SGLTs because it is not needed in this one. So let's see transporters of GLUT. So the first one is GLUT1, which is located in blood, blood-brain barrier and in heart. And GLUT2, it is located in liver, pancreas and small intestine. GLUT3, it is located in brain, neurons and sperm. And GLUT4, which is the main transporter for this glucose, it is located in skeletal muscles, adipose tissues and heart. And GLUT5, enterocytes of intestinal epithelium, which is luminal side only. So if you see, all these are non-insulin dependent, but GLUT4 is insulin dependent. All GLUT1, GLUT2, GLUT3 and GLUT5, all these four are non-insulin dependent. They don't need insulin for traveling of glucose. For example, we have seen in first one, liver, GLUT2, liver doesn't need any insulin for transportation of glucose. And blood, WBC, RBC, doesn't need anything. And blood-brain barrier, brain, no need. And heart, it doesn't need. So these all don't need any insulin for transportation of this glucose. They need only transporters to transport this glucose into the cell. So let's come back to our second point. So glucose is entered into the cell with the help of glucose transporter. So with the help of glucose transporter, it got entered into the cell. Now glucose is phosphorylated into glucose 6-phosphate with the help of glucokinase. So what our insulin will do in this step? Our insulin will increase the production of glucokinase. When it is increasing the production of this glucokinase, this glucose 6-phosphate, it is converted into ATP. The next function of this insulin is in liver and muscle cells, it will increase the synthesis of glycogen from glucose. And next one is insulin will also inhibit this glycogen synthase enzyme. When it is inhibiting this glycogen synthase enzyme, it will inhibit the glyconeolysis in liver. So the third action is it will decreases the gluconeogenesis in liver. So how it is doing? When we are taking any foods with fats, free fatty acids and with proteins and everything, it will go into the liver and it will inhibit the synthesis of phosphoenol pyruvate carboxykinase. When it is inhibiting this one, this glucose won't convert into carbohydrates or urea. So there won't be any gluconeogenesis. But if there is any deficiency of insulin, what will happen? All this proteins, amino acids and free fatty acids from peripheral tissue, they will go into the liver and there they will be converted into carbohydrates and urea. When they are converting like this is how in diabetic patients there will be more production of glucose, so that will cause us hyperglycemia. To avoid that, we need insulin. So the fourth point of this insulin is this insulin will inhibit lipolysis in adipose tissue. When it is inhibiting the lipolysis, it will favor the triglyceride synthesis. If there is no lipolysis, means there won't be any production of carbohydrates or any fats. But in diabetic patients, it will be in opposite action, because they are lacking of this insulin. So when there is lack of insulin, there will be more lipolysis due to some hormones like thyroxine and glucagon. So there will be increase of lipolysis. When there is increase of lipolysis, this will increase free fatty acids and glycerol in blood. Actually, in normal persons, this free fatty acids and glycerol is taken by our liver and it will produce acetyl-CoA. This acetyl-CoA is resynthesized to fatty acids and triglycerides. This is the normal function. But in this diabetic patients, these are taken by liver and it produces the acetyl-CoA, and this acetyl-CoA, instead of resynthesizing to this triglycerides or fatty acids, it will produce ketone bodies. So we know what will happen if there are more ketone bodies in our blood. Blood will become acidic in nature. Our body needs these ketone bodies, so they will be using this one in normal. But we have diabetes. So some of the ketone bodies are utilized by muscles and heart, but remaining they will go into the blood, and we know if there are more ketone bodies in blood, it will become acidic, our blood will become acidic and also causes ketoanemia and ketouria. So there will be more ketone bodies in urine and also in blood. So let's see the fifth point. So the fifth point is the insulin will increase the transcription of vascular endothelial lipoprotein lipase. So when it is increasing that, it will enhance the VLDL clearance and chylomicrons. So what is the function of this chylomicrons? It will carry all the fats to different parts of our body. So when there is a deficiency of this, there will be no VLDL clearance and also there will be no chylomicrons. When they are not there, when there is no VLDL clearance, there will be more production of fats, more production of unwanted cholesterol in our body. So when there is unwanted cholesterol in our body, that will causes harm to us. And next one is insulin will increase amino acid entry into the muscle. So when amino acids are entering into the muscles and other cells, they will utilize this for the synthesis of proteins. Okay, so this insulin will increase protein synthesis and as well as it will inhibit the protein breakdown. So when there is a deficiency of this insulin, their protein breakdown will increase and protein synthesis will decrease. When there is a decrease in protein synthesis, all the amino acids which will go into this blood, from blood, these amino acids are taken up by this liver and they are converted into pyruvate, glucose and urea. So excess amount of urea in our body is really very harmful to us. So they will result in negative nitrogen balance. So what is this negative nitrogen balance? So let's see first what is this nitrogen balance. So, and why do we need this nitrogen? We will take this nitrogen through our dietary sources like nuts, eggs, meat. So when we are taking this nitrogen is used for building of this proteins, building up of this proteins. So it is needed for our hair growth, nails growth and skin. So what is nitrogen balance? Amount of nitrogen we are taking and how much of nitrogen we are excreting from our urine. So this nitrogen is excreted through urine, feces and etc. So this negative nitrogen is the consumption of nitrogen will be less and excretion of nitrogen will be more. This will cause reduced nitrogen in our body and there won't be any buildup of this proteins, and not only that, we will be having more side effects of this. So then what will happen? Catabolism will be more and anabolism will be less. That will leads to diabetes. So all these mechanisms, all the 6 mechanisms will be happen within few seconds to few minutes. So let's see the mechanism of action of insulin.
How this is helping glucose to enter inside and what it is doing. So let's take any cell, skeletal muscle cell or liver cell. So imagine this is the cell membrane. So this is the insulin receptor. This insulin receptor consists of 4 subunits. 2 are alpha, which are located extracellularly and on transmembrane. So as this is on membrane, these are beta subunits, and to end intracellularly they are connected to tyrosine kinase. When insulin comes and binds with this will get activated. So this alpha, they both will come aside, like they both will attach with each other, okay, and they will activate this beta subunits. When beta subunits got activated, this tyrosine will get phosphorylated. So when this get phosphorylated, phosphorylation means activation, right? So it will do some kind of action. This will cause activation of insulin receptor substrate protein. So this substrate protein will do 3 functions. So first one, it will convert GTP into GDP, which will help GLUT4, which will help this GLUT4 to translocate on the membrane. So now GLUT4, which is inside the cell, will go and it will be on this membrane. It will go and it will stay on this membrane. So when it is going and locating on this membrane, this will help glucose entering into this cell. And the next function is, the next mechanism is, it will do phosphorylation or dephosphorylation of this phosphatidyl inositol 3 kinase. So whether it will phosphorylation or dephosphorylation. If it is doing phosphorylation, it will activate the enzyme. If it is doing dephosphorylation, then it will inactivate the enzyme. This IP3 kinase will also do another function, which is generation of PIP3, phosphatidyl inositol triphosphate. This will also generate this one. When it is generating this secondary messenger, it will also increase the translocation of GLUT4 onto the membrane. So even this will show the same mechanism, increase the entering of this glucose into the cell. So the third function of this IRS is RAS, regulation of cell division and differentiation. Here it will convert by converting GTP into GDP. This is converted into MAP kinase. So it will convert into MAP kinase. So what is this MAP kinase? Mitogen activated protein. So when it is activating this MAP kinase, DNA is transcripted to mRNA, which will regulate the proteins, which will help in the cell regulation and differentiation. So this is the mechanism of insulin. Once again, let's see the mechanism of insulin in detail. This is the insulin receptor which is located on the cell. It might be skeletal muscle or it might be any liver cell. So when it is located, this consists of 4 subunits. 2 are alpha subunits and 2 are beta subunits. Alpha subunits are located outside the cell and beta subunits, they are located transmembrane, which will be on the membrane. Inside, intracellularly, this beta subunits, they are attached with this tyrosine kinase. This tyrosine kinase, when they are inactivated, when there is no insulin, they are inactivated form. But once the insulin comes and binds with this insulin receptor, tyrosine kinase will get phosphorylated. That means it will get activated. So when it is getting activated, it will stimulate this insulin receptor substrate protein. So this will do 3 kinds of functions. So the first one is, it will convert GTP into GDP and it will activate GLUT4, which is inside the cell. So what is the function of this GLUT4? When this GLUT4 get activated, it will translocate it onto the membrane. That means from inside it will go and bind to this membrane. So when it is on the membrane, what it will do? It will get activated. When it is activating, it will allow more glucose to enter inside the cell. So that is the first function. And next function is, it will activate this IP3 kinase. So what is the function of that? When it is activated or phosphorylated or dephosphorylated, you will get, when it is phosphorylating, then you will have activation effect. When it is dephosphorylated, then it will show inactivated effect. So what is activating and deactivating? It is activating the metabolic action of the insulin. And the third function is, it will also activate RAS. RAS means regulation of cell division and cell differentiation. By converting this GDP into GDP, it will do MAP kinase. What is MAP kinase? Mitogen activated protein kinase. When this get activated, it will transcript DNA into mRNA. This mRNA is used for regulation of protein. This proteins, then it will regulate the cell division and cell differentiation. So this is the whole mechanism of insulin. So till when this insulin is attached to this insulin receptor, it will be for some time, and again when there is no glucose left here, it will go back to its normal place. But when we have glucose, till that time it will be onto this receptor only. So insulin, the max degradation happens in liver and least happens in vascular epithelium.
Neat. So when we are taking this nitrogen is used for building up of this proteins. So it is needed for our hair growth, nails growth, skin and everything. Okay. So what is nitrogen balance? It is the balance between how much amount of nitrogen we are taking and how much of nitrogen we are excreting from our urine. So this nitrogen is excreted through urine, feces and etc. So this negative nitrogen is, the consumption of nitrogen will be less and excretion of nitrogen will be more. this will cause reduced nitrogen in our body, and there won't be any buildup of these proteins, and not only that, catabolism will be more and anabolism will be less. That will leads to diabetes. So all these 6 mechanism will be happen within few seconds to few minutes. So let's see the mechanism of action of insulin.
So how this is helping glucose to enter inside and what it is doing. So let's take any cell, skeletal muscle cell or liver cell. So imagine this is the membrane, cell membrane. So this is the insulin receptor. This insulin receptor consists of 4 subunits. 2 are alpha, which are located extracellularly, and on transmembrane. So as this is on membrane, this is transmembrane. These are beta subunits, and to end intracellularly they are connected to tyrosine kinase. When insulin comes and binds with this will get activated. So this alpha, they both will come aside, like they both will attach with each other, and they will activate this beta subunits. When beta subunits got activated, this tyrosine will get phosphorylated. Okay, so when this get phosphorylated, it will do, phosphorylation means activation, right? This will cause activation of insulin receptor substrate protein. So this substrate protein will do 3 functions. So first one, it will convert GTP into GDP, which will help this GLUT4 to translocate on the membrane. So now GLUT4, which is inside the cell, will go and it will be on this membrane. It will go and it will stay on this membrane. So when it is going and locating on this membrane, this will help glucose entering into this cell. And the next function is, so the next function is, the next mechanism is, it will do phosphorylation or dephosphorylation of this phosphatidyl inositol 3 kinase. So whether it will phosphorylation or dephosphorylation. If it is doing phosphorylation, it will activate the enzyme. If it is doing dephosphorylation, then it will inactivate the enzyme. Okay, this will also do this IP3 kinase will also do another function, which is generation of PIP3, phosphatidyl inositol triphosphate. This will also generate this one. When it is generating this secondary messenger, it will also increase the translocation of GLUT4 onto the membrane. So even this will show the same mechanism, increase the entering of this glucose into the cell. So the third function of this IRS is RAS, regulation of cell division and differentiation. So what it will do, here it will convert GTP into GDP. This is converted into MAP kinase. So it will convert into MAP kinase. So what is this MAP kinase? Mitogen activated protein. So when it is activating this MAP kinase, DNA is transcripted to mRNA, which will regulate the proteins, which will help in the cell differentiation, cell regulation and differentiation. So this is the mechanism of insulin. Once again, let's see the mechanism of insulin in detail. This is the insulin receptor which is located on the cell. It might be skeletal muscle or it might be any liver cell. So when it is located, this consists of 4 subunits. 2 are alpha subunits and 2 are beta subunits. Alpha subunits are located outside the cell, and beta subunits, they are located transmembrane, which will be on the membrane. Inside, intracellularly, this beta subunits, they are attached with this tyrosine kinase. This tyrosine kinase, when they are inactivated, when there is no insulin, they are inactivated form. But once the insulin comes and binds with this insulin receptor, this tyrosine kinase will get phosphorylated. That means it will get activated. So when it is getting activated, it will stimulate this insulin receptor substrate protein. So this will do 3 kinds of functions. So the first one is, it will convert GTP into GDP and it will activate GLUT4, which is inside the cell. So what is the function of this GLUT4? When this GLUT4 get activated, it will translocate it onto the membrane. That means from inside it will go and bind to this membrane. So when it is on the membrane, what it will do? It will get activated. When it is activating, it will allow more glucose to enter inside the cell. So that is the first function. And next function is it will activate this IP3 kinase. So what is the function of that? When it is phosphorylated or dephosphorylated, you will get, when it is phosphorylating, then you will have activation effect. When it is dephosphorylated, then it will show inactivated effect. So what is activating and deactivating? It is activating the metabolic action of the insulin. And the third function is it will also activate RAS. RAS means regulation of cell division and cell differentiation. So in this, by converting this GDP into GDP, it will do MAP kinase. What is MAP kinase? Mitogen activated protein kinase. When this get activated, it will transcript DNA into mRNA. This mRNA is used for regulation of protein. This proteins, then it will regulate the cell division and cell differentiation. So this is the whole mechanism of insulin. So till when this insulin is attached to this insulin receptor, it will be for some time, and again when there is no glucose left here, it will go back to its normal place. But when we have glucose, till that time it will be onto this receptor only. So this insulin, the maximum degradation happens in liver and least happens in vascular epithelium.